Great. I'm going to go ahead and get started then. So I'm really grateful for this opportunity to introduce myself and present my program of research to you all. Wish I could be there in person. In today's talk, I'm going to first introduce you to the foundational concepts of psychosis that my research program is built upon, and then introduce you to the overarching goals of my research program. I'm then going to present a study that examined how structural MRI measures are associated with psychosis onset in an international cohort of participants at clinical high risk for developing psychosis. Then I'll show some more recent work where we combined multifactorial information from multiple brain regions to create a neuroimaging summary score to detect psychosis. And finally, I'll finish the study with the study we've been running that tests the feasibility of low field MRI to calculate these measures. Okay, so let's get started. The psychiatric syndrome I focus most of my research on is psychosis, which is characterized by a disruption in one's perceptions and thoughts, making it difficult to differentiate between what's real and not real. There are three main points that are really helpful for understanding the foundation of my lab's research: that psychosis typically emerges during adolescence and young adulthood, this really distinct period of brain maturation and specialization. We also know that psychotic symptoms occur on a spectrum, so it's fairly normal to walk down the street and mistakenly think, oh, someone's calling my name. But at the other end of the spectrum, and then turn around and, there's nothing. At the other end of the spectrum are full-blown hallucinations, such as hearing a voice distinct from your own that speaks back to you. Finally, I'll describe one of the cohorts I use to assess psychosis risk. This is a group that is called Clinical High Risk for Developing Psychosis, or CHR. And this group is most commonly characterized by individuals who are experiencing subthreshold psychotic symptoms, but they don't meet full criteria for a psychotic disorder. Approximately 20% of these individuals go on to develop psychosis, and they kind of fall within this subthreshold range, as I talked about. And most individuals are anywhere from age 10 to the early 20s, really encompassing that adolescent period. Our lab's research is grounded in the neurodevelopmental model of psychosis, which was originally developed as the neurodevelopmental model of schizophrenia in the 1980s, and it's really evolved over time. Broadly speaking, it proposes that psychosis is due to a number of deviations from typical neurodevelopment, such as excessive synaptic pruning, reduced interneuron activity, and/or deficient myelination. And we can't directly assess these processes in humans, but neuroimaging is the closest in vivo measure of brain structure and functioning. So what myself and others have been doing is harmonizing neuroimaging data across a wide age range and characterizing normative development to really serve as a foundation for understanding atypical development. And most graphs I'll be showing you today will have age on the x-axis and some sort of neuroimaging measure on the y-axis. And once these normative trajectories are established, you can then examine, well, how do clinical samples deviate from typical neural development? And this process is really akin to growth charts for height and weight, and to making sure that consistent and normative development is occurring. This happens with pediatrician offices all the time. By comparing a child's information to a large reference group, if a child stopped growing for some reason, you'd know there was a problem and intervene. And this approach has been termed normative modeling, and there have been some really fabulous collaborations combining large amounts of data to show this age-related neurodevelopmental change across the lifespan. And so by bringing together these large-scale collaborations focused on clinical high risk for psychosis, individuals at clinical high risk for psychosis, or CHR, using novel methods to improve neuroimaging's ability to differentiate those with psychosis from those without, and couching that within a developmental framework, we're then working to test out low-cost neuroimaging measures to make them MRI accessible and affordable for all. And that's the story I'll be sharing with you today. I'll move on now to the first study. In 2009, 2010, Paul Thompson at USC created the Enhancing Neuroimaging Genetics Through Meta-Analysis, or ENIGMA Consortium, where one of the main goals of this collaboration is to harmonize neuroimaging data and identify neural markers of psychiatric illness. And I really think it's at this way, by combining data from all across the world, we're able to really get at the ground truth for detecting group differences in psychiatric illness. So in 2018, with Dennis Hernas at Maastricht University, we started the Clinical High Risk for Psychosis Working Group. And since then, we've brought together and harmonized neuroimaging and clinical data from more than 31 different sites. And I'll be presenting on the first study of these results today. Now, despite years of research within the CHR field, when we started this study, or when we published this study, there was really inconsistent evidence for the ability of neuroimaging measures to predict psychosis onset in this sample. And that's likely due to a combination of factors. It's a really heterogeneous sample. So studies often lack power to detect real effects because they're these small individual studies. The effects are likely more subtle because it's very early in the course of illness. And then most neuroimaging studies of CHR participants don't look at the effects of development. So they might be undermining or obscuring important effects. So with this large, well-powered sample, we can address these issues. And so for this first study, we ended up with over 1,300 healthy controls and approximately 1,500 CHR individuals with follow-up data. Now, it's important just to clarify the study design here. At the baseline assessment, that's when the participants are first ascertained in the study. No one meets criteria for a full psychotic disorder diagnosis. The individual sites conduct a clinical assessment that determines, yes, this participant qualifies as high risk, and then the participant completes an MRI scan. The CHR participants are then followed over time and assessed again, and that's when what we call conversion status, whether they develop a psychotic disorder or not, is assigned. And when the participant had the MRI scan back at this baseline assessment, we didn't know whether they would fall into the converter group or the non-converter group. So whether they developed a psychotic disorder versus whether they did not. It's only later on at the follow-up assessment that we assign group status. So this really allows us to look at prediction. Because we're combining data from all over the world, we need an MRI scan that's common to collect. And so we focused on T1 weighted gray matter structural neuroimaging. For the cortex, we used the standard atlas parcellation available and measured cortical thickness and surface area. I'll be showing you sagittal plots of the medial and lateral views today. I'll also show you some subcortical volume findings in the coronal view. So the first question we asked was, can we use imaging to predict which of these high-risk individuals will transition to a full-blown psychotic disorder? And we ran general linear models looking at the difference between healthy controls, CHR converters, and CHR non-converters. And I'll be showing you the plots for cortical thickness on the left, surface area in the middle here, and then subcortical volume on the right. And for cortical thickness, for these all different measures, I'm showing you the left and the right hemisphere. This top row just refers to the F statistic, which just says, hey, is there any overall effective group? And our group variable is CHR converters versus CHR non-converters versus healthy controls. So the darker the purple, the stronger the effective group. And just visually, you can really see all the dark purple falls in the area of the cortical thickness. That's where all the group differences are. And it's important to point out that where you see group effects are just as important as where there are not group effects. We don't see a lot of group effects in surface areas of cortical volume, which might give us some insight into some of the mechanisms underlying psychosis onset. Now, in any region that was still statistically significant after correction for multiple comparisons, we conducted these post hoc comparison analyses between two groups at a time. So first here, I'm showing you the comparison between CHR converters and healthy controls. And here I'm now showing the Cohen's d effect sizes, which basically the colors reflect the strength of the effect size of the group comparison, with brighter red indicating that healthy controls had greater cortical thickness compared to CHR converters. You can also do this for CHR converters versus CHR non-converters. So those high-risk kiddos that went on to develop a psychotic disorder versus the high-risk individuals who did not develop a psychotic disorder. And most researchers' primary interest is what regions differentiate both the CHR converters from healthy controls and the CHR converters from the non-converters. And what we saw is that there were cortical thickness reduction in 3 regions: the paracentral lobule, superior temporal cortex, and the fusiform region. And these bar graphs now just visualize the 3 cortical thickness measures of all 3 groups together. So this is, you've got the CHR converters in pink and the non-converters in purple, which fall within the middle, and then the healthy controls over here in teal. And what's interesting to me, I think, is that 2 of these regions are considered functionally relevant for perceptual positive symptoms. That's the fusiform region and the superior temporal region. The fusiform is involved in higher order visual processing, and the superior temporal cortex is involved in auditory processing. And both of these regions have been previously implicated in schizophrenia. So you might wonder, well, are we starting to get at, do we have a biomarker to identify psychosis before disorder onset? However, it's important to note group difference was only associated with 2 to 4% of the variance in these measures. So really, it's impossible at the individual level to differentiate those who develop a psychotic disorder from those high-risk individuals who do not, with that small of an effect size. So we then asked, well, what about the role of development in these regions that we see group differences? And we assessed the interaction between age and group status using generalized additive mixed models for these 3 regions. And for the purposes of this talk, I'm going to focus on the age-associated disruptions we observed in cortical thickness in the paracentral lobule. So what we saw with healthy controls here in teal is this characteristic decrease in cortical thickness with a slowing of growth in the early 20s. And this has been repeatedly shown in many developmental neuroimaging studies. And for these plots, I'm showing you the line of best fit, and the light shading around it is the standard error. And when you present it this way, this gives you an overall pattern, allows you to capture the overall pattern of neurodevelopmental trajectories. I should say that when you look at the individual data points, there is a fair amount of variability. And then in these plots underneath, what I'm showing you is the derivative or the rate of change for each group separately. So the healthy controls, the non-converters, and the converters. And what darker blue indicates is a period of greater decrease. So for example, you can see that the healthy controls are showing this steady decrease with a sharper decrease in right before their 20s and then plateauing out. And you see a similar pattern of effect with the clinical high-risk non-converters, as observed by this purple line. But what's striking is that the CHR and converters do not show the accelerated decrease early on. They have a flatter, more shallow slope in late childhood and early adolescence. And then during the late teens, early 20s, the CHR converters are still showing a significant decrease while these other 2 groups are plateauing off. And so they're showing a steeper slope when these other individuals have sort of slowed in their growth. Now, what I think is really super cool is that since this study on the right that I just showed you was published in 2021, I've worked with Carrie Bearden, I think she presented yesterday, to examine another high-risk cohort, 22q microdeletion syndrome, a genetic disorder that confers significantly increased risk for detecting psychosis on the left. And what we found here, this was across the entire brain, where we only found it in one region, is that you're starting to see some convergence on these age-associated effects, with the shallower slope in those with psychosis spectrum symptoms, seeing that in two different high-risk groups during late childhood, early adolescence, and then the steeper slope of cortical thickness change in late adolescence, early adulthood. So this might provide some suggestive developmental mechanisms responsible for psychosis onset. So maybe the first age-associated alteration marks these individuals with earlier onset and more severe course. Or perhaps this second age-associated aberration is indicative of a later onset group that's driven by differing neurobiological mechanisms like synaptic pruning or inflammation. So for the last theme of the study, we wanted to know, well, how do our findings compare with other existing Enigma working group studies? And so we have this harmonized neuroimaging data, so we can take advantage of this and compare our results across different studies. So for every study, we have the same measures of same neuroimaging measures. So on this graph, each dot on the graph reflects the effect size associated with one of these brain regions. And so this, on the x-axis here, these are the effect sizes for the group difference between schizophrenia versus controls. This was a publication by Theo van Eert in 2018 or 2017. And you can then say, well, what is the correlation with what we found, the group difference effect sizes in our study? And what we see is a positive correlation between the two groups. So for both studies, the strongest effect sizes were within specific temporal and prefrontal regions. And so what does this mean? It means that the regions that we obtained the strongest group differences were some of the strongest group differences in independent study for adults with schizophrenia. And we also found this when we compared Frank Sun's and Carrie Bearden's work of individuals looking at individual 22q microdeletion with psychosis versus 22q microdeletion without psychosis compared to our effect sizes. And so it's much more, it's not about, oh, it's just this one region that's affected. It's much more to me about the pattern of abnormalities that you're seeing. And I also think it was striking that this convergence across three different groups really provides strong scientific evidence that we're tapping into something real with these cortical thickness abnormalities. So in conclusion, because these abnormalities were only found in cortical thickness measures, you can speculate about several different neurobiological mechanisms that might underlie these abnormalities. So thinner cortical thickness could reflect abnormal synaptic pruning or alterations in myelination. The age-associated findings might suggest other possible mechanisms. So earlier onset might reflect deterioration of this neural circuitry, or maybe there's this accelerated developmental decrease before we measured the individuals. Or if we think perhaps of adolescence as this time of specialization, where you must go through this change in order to, during this particular time, to reach optimal adult levels, we might be seeing a lack or delay of this specialization in those who develop psychosis. Finally, the overall group differences were robust but small. And so I really think it's, again, we're focusing on this overall pattern. And then I think what is probably the most exciting to me is that Dennis and I have created this resource for many different investigators to use. And we've had several additional publications since 2021 and have about 7 other ongoing projects right now. So using that information, we then started to think, well, how can we leverage this data from these large-scale studies of psychosis? So, as I said earlier in the study that we found, group status affects for only 2 to 4% of the variance in these individual neuroimaging measures. And we wanted to find a more robust way to detect neural signal associated with psychosis group status. And so through discussions with my K-mentor, Bernie Devlin, we started thinking about how these adult studies of schizophrenia have really provided us with this ground truth of what's disrupted in schizophrenia. And we have this converging evidence from multiple samples that it's not about one or two brain regions, it's the pattern. So we thought, well, how can we use this information? And in this project led by Amanda Rodrigue in David's Galan lab, we assessed 3 samples of adults with schizophrenia and healthy controls with MRI data. We extracted structural neuroimaging measures as well as diffusion-weighted imaging measures, which measures the large white matter tracts in the brain, weighted these individual values by the Enigma Schizophrenia Working Group effect sizes, and then we summed the score across the entire brain to create a single schizophrenia neuroscore. We calculated a score for T1 weighted structural MRI, diffusion weighted MRI, and then a multimodal score that was a combination of both. And for those of you who do genetic work, this is analogous to a polygenic risk score for a particular phenotype. But in this case, a higher score putatively indicates greater neuroanatomic liability for a particular phenotype. And so we combined all of these 3 samples and conducted a mega-analysis of the group effects. And we found that individuals with a schizophrenia diagnosis had greater neuroscores in comparison to controls with the T1 neuroscore, the DTI schizophrenia neuroscore, and the multimodal neuroscore. Now, the effect sizes are a bit larger, and the finding was consistent across different study samples, which is not the case for individual regions. And importantly, these effect sizes are significantly larger than any of the individual regional case control group differences. We then found out for all the neuroscores, lower IQ here on the x-axis was associated with a higher neuroscore. So lower IQ associated with this putative greater liability for neuroanatomic liability for schizophrenia. And we also found this in not only in schizophrenia individuals, but in healthy controls, which is important for understanding that this score may not just relate to schizophrenia, but also individual differences in behaviors. Then for the T1 and the multimodal score, but not the DTI score, higher positive symptom severity was associated with a greater score. And significant correlations between positive symptoms in individual brain regions are often scarce. And even when they are identified, the relationships are often small, accounting for less than 1% of the variation between the 2 measures. But in this study, the T1 weighted score accounted for about 4% of the variance in positive symptoms, suggesting that these neuroscores may be a more robust index than measures of individual brain regions. Finally, only the T1 weighted neuroscore here on the left was associated with negative symptoms, with higher negative symptoms associated with the higher score. So in summary, these findings show that the neuroscores are important for more than just detecting group differences. So in conclusion, neuroscores are an important step forward in identifying these robust neurobiological markers of disease. And these summary scores and similar ones like it, as this is a very active area of research, may enhance our understanding of psychiatric disorders and individual differences in behavior. And finally, because we saw this effect in multiple samples, we are starting to get some solid evidence that this effect is generalizable. Now, that study was on adults, and as I mentioned at the start of my talk, I'm really interested in detecting risk in youth. So now, as the continuation of this work, by bringing together those many archival neuroimaging data sets of typical development, you can plot the developmental trajectory of the neuroscore. So with increasing age, we see that the T1, the schizophrenia neuroscore, increases with increased age. And if we account for that variability here, we may improve our ability to detect youth at greatest risk. You can look at an independent set of youth maybe obtain this with an individual with the orange scale, and then obtain a deviation index, the red dot, with greater deviation from the age-relevant score to serve as the more appropriate indicator of risk. So we will then, after detecting this deviation, we can then weight that information by the effect sizes and create a deviation neuroscore. And so to do this, we're taking advantage of some of the wonderful normative modeling findings that have established cortical thickness trajectories at the population level and made them publicly accessible. So stay tuned for that work. In the last portion of my talk, I wanted to talk about a newer direction that I've been taking my work since I came to Boston Children's about 2 years ago. I started thinking, well, once we have this successful neuroindicator of risk, how do we use the tool in an accessible, scalable way? So MRI scans are very expensive. People must come to a specialized center. There's a lot of safety concerns associated with it. And research participants are often samples of convenience. So we really need to find additional ways to reach underserved communities. And it's also been shown through multiple other instances of pediatric care that risk factor measurement is most effective when it's performed in a community clinical setting. So this suggests that our typical research grade 3T MRI scanners, the measurement of that is limited for the masses because, you know, those of you who use 3T know that it's in a very specialized center. There's lots of training, strict schedules. And so what's been exciting is that there's this newly developed 64 millitest, a low field scanner by Hyperfine that might be our answer. It's cheaper. The installation is much cheaper. The maintenance is cheaper. It's portable. A research assistant can be trained to administer this scan. You don't get the artifacts from braces that you get with kiddos. And there are reducing a lot of the anxiety concerns that come with going into an MRI. And so we're conducting a 5-year study where we aim to look at 200 typically developing youth. And this is the low field scanner. This was probably like one of the best days of my life. I was so excited to get it. And what we're doing is we're examining the relationship between the schizophrenia neuroscore generated in the 3T scan to the schizophrenia neuroscore generated with the low field MRI. And this work led by 2 ladies in our lab, Mary Corcoran and Rebecca Hayes, is that what you can do is, it's some mathematical magic basically, is you see this, this is the individual of a 3T scan of a youth, okay? And then you can see the low field scan, which is basically a much more poor resolution of the 3T version, right? Now, what's fascinating is if you look over here on the right, is that there are these super resolution approaches, which basically use machine learning techniques such as convolutional neural networks to improve image quality and resolution within the low field acquired systems, the low field acquired systems. And this method is called SynthSR. And when you apply SynthSR, you improve the quality of the image. As you can see here, this is the image that was originally started over here, and it looks much more like the 3T measure. So over the last 1.5 years, we've collected 3T and low field scans and processed and analyzed this data on 70 young people. And you can see that the sample is pretty evenly split across self-reported biological sex. there's a fair amount of diversity with within our sample. It's 60% white, 10% Hispanic. And what happened was every individual received a 3T MRI scan session where they received a T1 weighted scan and a T2 weighted scan. And then we processed that data through FreeSurfer. Either on the same day or within a week of receiving that scan, they also participated in a low field MRI scan session. Now, to improve for signal to noise ratio, what you can do is collect multiple scans in multiple orientations. So we actually ended up collecting 6 sets of scans for the one T1 weighted, one T2 weighted, in each direction, each orientation, axial, coronal, and sagittal, then co-registered these scans, then applied it SynthSR, and then used FreeSurfer. And this is showing the correlation now of the low field measures with 3T measures. Now, this is with minimal processing and not with the repeated scans. And so the darker the red, the stronger, the more positive the correlation. So you do see that in cortical surface area and cortical volume, there's widespread moderate correlations between the 3T measures and the low field measures. Now, what's fascinating is that when you apply SynthSR to the low field measures, you can significantly improve the correspondence between the low field measures and the 3T measures. As you can see that the red is getting stronger here for the correlations. We then use statistical z-tests to compare, well, is this method better than this method? Are the correlations obtained in this method stronger than the correlations obtained in that method? And if it's in the darker the red, the stronger this improvement. And so what you can see across cortical volume and cortical surface area measures, as well as subcortical volume measures, and distinct temporal and medial regions of cortical thickness, you can improve this correspondence, suggesting that, yes, we should be able to have some ability to measure these scores in a portable low field scanner. So basically, hopefully today I've shown you that these large international collaborations will help us understand mechanisms of and risk factors contributing to psychosis, that a developmental framework will improve our understanding of risk factors contributing to psychosis, that we can use these novel methods to harness the power of neuroimaging to detect psychosis, and that low field MRI is a promising technology for measuring accessible and cost-effective biological risk factors. And with that, I'd like to thank you, most of all, the Enigma working group members. The first study I would not have been able to do without the collaboration of more than 150 individuals. Dennis Hernas at Maastricht University co-chairs the group with me. David Glahn is my mentor, and David Glahn and Bernie Devlin have been wonderful mentors. And then there's a whole host of other individuals who have contributed to this research. I'm just one of many working to move this forward. And then finally, the participants and their families and my funding sources, I'd like to thank.